So hello everybody and welcome to the Metrion Biosciences Ion Channel in Drug Discovery webinar series. So our webinar today is going to be presented by Dr. Greg Carr from the Lieber Institute for Brain Development. We're delighted to have Greg with us today, and the title of his presentation is A Role for hERG Potassium Channels in Cognitive Function. So just a brief description of how to ask a question if you'd like to during the presentation. So please do feel free to ask questions at any time using the GoToWebinar control panel. There's a questions tab, which you will see towards the bottom, well, towards the middle of the panel there, just above the handouts and the chat. Please feel free to ask any questions in the question box and any technical issues anybody's encountering, please pop this in the chat and we will address them. So all of your questions will be answered at the end of the presentation by Dr. Carr, and Eddie Stevens, my colleague who's joined me here today, will run the Q&A. So please note that the event is being recorded and the presentation will be made available post-event. So just for anyone who's not aware of Metrion, I'm going to provide just a very brief overview of what we do. So Metrion Biosciences, we're a preclinical CRO. We're located here in Cambridge, UK at Granta Park. We recently moved into a brand new building here, which is fantastic. There's a photo of it just from there. And we specialise in high quality ion channel drug discovery and safety pharmacology screening services. So we're a specialist ion channel service provider. Ion channels really are our bread and butter. We've got proven electrophysiology expertise. We offer high quality ion channel assays on either an FTE or a fee-for-service basis. We have an industry-leading panel of in vitro cardiac ion channel safety assays, and we also offer translational native cell and phenotypic assays for both neurological and also cardiotoxicity testing. So we also offer cell line development and cell line optimization services, and we offer tailored assay formats, data analysis and reporting solutions for effective project management and quality assured data packages. So Metrion offer independent, cost-effective and flexible services to meet all the customer needs. And please feel free to get in touch with us after the webinar if you have any questions regarding our services or would like to request a quotation. So I'm now going to hand over to my colleague Eddie Stevens, who will introduce Greg. Thanks, Eddie.
Right. Good afternoon, everybody. So I'm really pleased to introduce Dr. Greg Carr, who's lead investigator in the Drug Discovery and Development Group at the Lieber Institute for Brain Development, and also assistant professor in Johns Hopkins University School of Medicine. So Greg leads the in vivo pharmacology laboratory in the Lieber Institute, with the goal of identifying novel therapeutics for the treatment of neurological and psychiatric disorders. So prior to joining the Lieber Institute, Dr. Carr was a postdoctoral fellow at the Clinical Brain Disorders branch of the National Institute of Mental Health and an adjunct instructor for the Department of Biology at Howard University. So Greg received a BA in neuroscience from Kenyon College and a PhD in neuroscience from the University of Pennsylvania. So, okay, thanks very much, Greg. So feel free to start whenever you're ready.
Thank you, Eddie, for the kind introduction, and thank you, Eddie and Sophie, for the invitation to present here today. So I'm going to get my screen together. So hopefully you are seeing my slides.
Yeah, I'm seeing the slides.
Okay, perfect. I'm really excited to talk about our work on this particular project. So we've been working on a drug discovery program focused on targeting the hERG potassium channel. But before I get into the details of our work on hERG, I want to give a brief introduction to the Lieber Institute and the work that we do, and specifically the strategies we employ to accomplish the mission of the Institute, and tie that into how we became interested in working on this particular project. So the Lieber Institute for Brain Development and the Maltz Research Laboratories is a nonprofit research institute focused on, one, improving our understanding of the underlying causes of schizophrenia and related disorders, and two, developing novel therapeutics to treat these disorders. It's an interdisciplinary group of researchers. Our main facility is in Baltimore, Maryland, on the Johns Hopkins Medical Campus. And we also have offices around the country and relationships with medical examiners' offices in California, North Dakota, and Michigan. So we focus on three big questions as it relates to brain research. So the first is, how do genes contribute to risk for psychiatric illness? Second, how do environmental factors contribute to risk as well? And then finally, how can we use that information to study factors and that lead to the development and improvement of therapeutics? So the interdisciplinary group of researchers we have at the Institute allow us to take a comprehensive approach to developing therapeutics. So we can bring in information from postmortem human brains, clinical genetics, clinical pharmacology, and identify genomic and transcriptomic signatures associated with the disorder to inform the generation of cell-based and animal models. And we hope this will lead to fruitful drug discovery campaigns that ultimately can improve the lives of patients. And so this story I'm going to tell about the hERG channel and our work with it, I hope will demonstrate the power and utility of this technique. And so leading into that, I'm going to give a brief introduction to the public health metrics associated with schizophrenia. So roughly 0.5 to 1% of the population is estimated to have schizophrenia. This is around 3 million people in the United States. Onset is typically in late adolescence and early adulthood. And it's characterized by three categories of symptoms. These are termed positive, negative, and cognitive symptoms. The positive symptoms are really the hallmarks of schizophrenia. They include hallucinations and delusions. They're sometimes referred as a collective as psychosis. The negative symptoms are characterized by a loss of function. So anhedonia, loss of pleasure, lack of movement, negative affect. The cognitive symptoms include impairments in attention function, working memory deficits, slowed processing speed. There's also quite a bit of morbidity and mortality associated with the disorder. About 5% of patients will die by suicide. And even taking suicide out of the equation, the disorder is associated with a roughly 20-year decrease in life expectancy. There are also some significant gaps in the treatment landscape. So about 50% of people do not receive treatment, diagnosed patients. And the currently available treatments do an okay job of suppressing positive symptoms, but they're not very effective against either the negative or cognitive symptoms of the disorder. So this is really unfortunate because the severity of the cognitive symptoms is directly correlated with functional outcome and quality of life for patients. So there's a clear need for new therapeutic options. And in our lab, we focus on the cognitive symptoms because we believe this is the area of greatest need. So almost 12 years ago now, when I started my postdoc at the NIH, this paper had come out from the lab that I was just joining, and it was a really hot topic in the lab. So it described the discovery of a unique transcript of the KCNH2 gene, which codes for hERG channel subunit or KV11.1. There are a number of different names for this particular gene product. So interestingly, they found that this isoform is more abundantly expressed in brain when you compare to expression in the heart. And it seems to be relatively new from an evolutionary perspective, as it's only found in primates. What they also found was expression of the isoform affects neuronal function and behavior, and is also associated with cognitive impairment in the context of schizophrenia. So one of the first things that they noticed was that the SNPs in the gene were associated with altered fMRI activity in the hippocampus and dorsolateral prefrontal cortex when subjects were engaged in neuropsychological testing. And so we'll see here the T allele is what's referred to as the risk allele. And so we'll see a bit more data surrounding this. So first, T carriers have higher expression of this novel isoform, which from now on we'll call 3.1, and is decreased expression of the full-length canonical 1A isoform. So in this cohort, there was significant interaction with diagnosis, as patients with schizophrenia had an even higher level of expression of 3.1. So the isoform was sequenced, and it turns out it's a truncated form with deletions within the PAS domain. So the next question was, does this change in sequence and structure produce functional effects? So I'll take a moment to walk through the channel kinetics here. So the KCNH2 channel is responsible for a delayed rectifier current, or IKR, that's critical for repolarization of the cardiac action potential. So it's known as a fast inactivating, slow deactivating channel. And you can see what that means here functionally here. So in response to a depolarizing event, the channel opens and some current flows, but it quickly moves into an inactive state. It then transitions from inactive to open and slowly transitions back to the closed state. This gives you sort of the proper repolarization, and you can see why disruption here would potentially lead to the arrhythmias that are often found with mutations in this channel or blocking this channel in the heart. So when expressed in HEC cells, what we see is with the 1A isoform, the canonical isoform, depolarizing events lead to channel opening and a slow return to the closed state. And so the question is, how does the 3.1 isoform respond here? So 3.1 is what we refer to as a fast deactivating channel. Because of this, it doesn't conduct nearly as much current as 1A, but you can see that both 1A and 3.1 are sensitive to the non-selective hERG blocker E4031. So when expressed in cortical neurons, the result is an increase in the basal firing rate compared to control neurons. This makes some intuitive sense because incorporation of 3.1 subunits seem to allow the cell to return to its resting membrane potential faster, and therefore it can fire the next action potential sooner. So in the first part here, we've seen that there's a particular isoform of the KCNH2 channel that's expressed at higher levels in the brain than in the heart. It's also, I didn't show this data, but it's also about 1,000-fold less abundant than 1A in the heart, so very lowly expressed outside of the brain. The association of 3.1 expression with impaired cognitive function was replicated in an independent study done with a separate cohort of patients. And as far as ion channel properties, the 3.1 isoform is a fast deactivating channel with respect to 1A. And in neurons, this produces an increase in the basal firing rate. So what was needed at this time was a model system to determine what happens in an intact animal. Unfortunately, as I said earlier, 3.1 is only expressed in primates. And on top of that, what we needed was a model that would give an overexpression phenotype. So right before I joined the group, Jingshan Chen had designed a double transgenic mouse that overexpressed the 3.1 isoform selectively in neurons. So he used what's called a TET-off inducible system, where the 3.1 transgene was expressed under the control of a neuron-specific enolase promoter driving the tetracycline transactivator. And so with this system, we can turn off expression of the transgene with administration of a tetracycline antibiotic. In our case, we used doxycycline. And so with this in hand, me and Feng Yang, electrophysiologist with the group, were tasked with phenotyping this mouse. So very quickly, Feng found that the 3.1 isoform had effects in the mouse similar to what had been shown in primary neurons. So it increased the basal firing rate. And as you can see, this increase sort of scaled linearly with the current injection. On the behavioral end, I was interested in tasks that engage the two brain regions where dysfunction had been seen in patients, so specifically the hippocampus and prefrontal cortex. A task that we know engages both regions and in fact has been shown to depend on synchronized activity between the regions is a spatial working memory task called the Discrete Paired Trial Variable Delay T-maze. This is one of a family of tasks referred to as delayed non-match to place tests, or DNMTPs. So I'll walk through here how this task is structured. First, mice are placed into a T-maze and trained on the non-match to place rule. The discrete paired trial component is this. In the first run of a trial, one of the arms is blocked. The mice travel down the maze and go down the open arm and retrieve a food reward at the end. The non-match to place rule is that in the next run, which occurs 4 seconds after the first, both arms of the T are open, but only the arm that was previously blocked has the food reward. Mice learn that on the second or choice run, as we call it, they should go to the opposite arm from the forced run. We train them on this for a few days with 15 minutes between each trial, and eventually they get really good at completing this task. You can see here that both control littermates and the 3.1 transgenic mice learn the rule, and if anything, the 3.1 mice acquire the rule slightly faster than controls. The parameters during training are relatively easy. With trials 15 minutes apart, there's very little interference from previous trials. What I mean by that is if we think of working memory as a sort of scratch pad, you only keep information online as long as you need it to make decisions. So once information is no longer needed, you can discard it. One way to tax working memory and really get at this is to increase the number of trials per unit time. When we do this, we see the performance of the 3.1 mice decrease significantly. When the ITI goes from 15 minutes to 15 minutes to 20 seconds, and we vary the intra-trial delay, they perform significantly worse than control mice. So they're having difficulty keeping the relevant information online. So we were really excited to see this result, but we were also looking for other behavioral deficits in this model, particularly ones that would be more amenable to therapeutic intervention. So the T-maze is great, but due to the timing of the trials, each session being over 4 hours long, and training occurring over multiple days, it's often difficult to combine the pharmacological interventions. So we next tested the mice, and in another test I thought we might see deficits, given the critical role of the hippocampus, and that's the object location test. Here again, a very simple task. Mice are given 10 minutes to explore 2 identical objects in an open field, and 1 hour later, they're put back into the arena with the same objects. The only difference is one is displaced. All other things being equal, and there are a series of controls we run to ensure that's the case, mice tend to explore the object that's been moved more than the one in its original location. And you can capture that in either a discrimination ratio or index, where a 0 value means the mice show no preference and a positive value indicates the mice spend more time or show preference for the displaced object. So we found that control mice show the expected preference, while the 3.1 mice show no preference. Interestingly, 3.1 mice show normal novel object recognition and preference, where instead of moving an object, you replace it with a novel object that they haven't interacted with before. So the results here in object location aren't the result of the animals having an aversive response to objects or not liking to explore. So here we have a very clear picture of the effects of the transgene in vivo. But there's a big issue for us when it comes to modulating this channel, right? These mice have overexpression of 3.1 their entire lives. So we don't know if these phenotypes we're seeing are the result of acute effects of the channel, which should be amenable to pharmacology, or whether the result is from a diffuse set of adaptations or compensation plasticity that occurs over the lifespan. So as a partial test of this, we fed a cohort of mice doxycycline for a few weeks during adulthood to turn off expression of the transgene. And what you'll see is that turning off expression here in adulthood reversed the object location phenotype. So this gave us some hope that there may be a way to correct these pathological phenotypes in adulthood, and thinking translationally, it may be possible to treat patients in the clinic. And so with that, Fung continued to test the 3.1 mice in a number of ePhys models as well. And he found some interesting phenotypes, one being impaired theta burst stimulation, LTP, in the hippocampus. They had normal high frequency stimulation, LTP, so it's not as if the circuit was completely inactive. It just seemed to be less sensitive to input than in control mice. We gave Fung some of our dox-treated mice as well. And sure enough, he found that doxycycline treatment was able to rescue the theta burst LTP phenotype. Again, showing that this may be responsive to acute activity or lack of activity in the 3.1 isoform. So not to give you the impression that these mice are impaired in every sort of task that we test. In fact, the supplemental materials for the molecular psychiatry paper here has about 2 years worth of behavioral data with no significant effects. So I want to show some interesting data that we haven't published yet that led us to an interesting line of work with these mice. So we tested the 3.1 mice in a task that measures attention and impulsivity called the 5-choice serial reaction time task. Here, mice are in an operant chamber and in front of a 5-location array. Each trial, one of the 5 locations is illuminated, and if the mouse nose pokes the location where the light appears, it receives a food reward. I have to say I was shocked when the 3.1 mice performed this task just as well as control mice on just about every measure. However, there was one component of the behavior that was a little odd. They were consistently a split second slower to retrieve their reward than controls. So in rodents, this reward latency is a sensitive measure of motivation. The more motivated the mouse, the faster they move to get their reward. So I thought this was interesting. So the mice were motivated enough to perform the task at a high level, but they just weren't as excited to retrieve this future reward. And so this finding made me think of a paper from James Gold and colleagues who were across town here in Baltimore at the University of Maryland, showing that one of the hallmarks of schizophrenia is altered reward processing and specifically steeper discounting of future rewards. So we decided to test this directly by asking the mice, how much are you willing to work for a future reward? When we keep it simple and put the mice on a low fixed ratio schedule where the mice can actually predict when they will get a reward, we see no difference between the 3.1 mice and their controls. So here, fixed ratio 1 and fixed ratio 3 mean that after each one, either 1 nose poke or 3, the mice will receive a reward pellet. When we make things a bit more complicated by moving to a progressive ratio schedule, we found something I think very interesting. So in a progressive ratio test, each subsequent reward requires a greater number of nose pokes than the previous one. So for this, we use an exponential formula for a couple reasons. One, so that the number of nose pokes required for a reward would increase quickly, and also so that the mice couldn't predict the number of responses that would be required to get the next reward. So here with the control mice, we see exactly what we would expect. First, mice are willing to work more when they are food restricted compared to when they have free access to food in their home cage. You see that both in the number of rewards earned and in the breakpoint. So breakpoint really is just the highest number of responses the mouse is willing to make for a single reward before giving up on the task. You can see for food restricted mice, it's about 70 responses, and when they're less hungry, it's only 40 or so. So what we see in the 3.1 mice is they are less willing to work for food reward than control mice. And it doesn't matter whether they're food restricted or not in terms of an interaction with the effect. It's really a linear effect across conditions. So the motivational deficit we saw in the 5-choice holds up here in a more formal test of motivation. So really just to summarize the behavior, 3.1 mice have disrupted spatial working memory, object location memory, and motivational deficits. In the spirit, though, of this seminar series being ion channels in drug discovery, I'm going to spend the rest of the talk here talking about a strategy we've implemented using this behavioral platform I've described to test pharmacological intervention, right? The strategy we chose, given the data on the effects of taking 3.1 offline with doxycycline, is a 3.1 specific inhibitor strategy. So yes, we are intentionally inhibiting the hERG channel, and we think that'll have some therapeutic benefit. So granted, as I've explained here, the 3.1 isoform is not highly expressed in heart and is not a critical component of the cardiac action potential. So we don't think selectively targeting 3.1 will have sort of dangerous side effects. The difficulty really is, how do you get selectivity with not a huge difference in the structure of the two isoforms? And so through the efforts of a really talented grad student in the group, Nick Calcaterra, we've had some success getting selectivity in compounds. So we have a tool compound here, LI507, that has good potency and about a 10-fold selectivity for 3.1 over 1A. It's not a huge window, but since 1A is not a key component of the mouse cardiac action potential, we can push the dose if needed for our pharmacodynamic assays. We took our first shot at this with the object location test. So we dosed mice with LI507 and timed the dosing so that the entire object location test would be within this nice 2 to 3 hour window that occurs 2 hours post-dose, where we have relatively stable plasma levels of the compound. And so what we see is the 1 milligram per kilogram dose produced a significant preference, and it was significantly higher than vehicle treatment here in object location. So for us, this was a nice proof of concept that these compounds can work in vivo, but there are a few features that are less than ideal here. One relates to the task. You can only test mice in object location once, and so you're limited on the amount of information you can get on repeated administrations. The other was compound related. So 507 doesn't have the best physical properties. And so we'd like to move to a more soluble compound so we don't have to use corn oil, which the mice tolerate fairly well, but there's some better vehicles, we think. Luckily for us, we do have a better tool compound. LI967 is available for testing now. It has similar potency on 3.1 compared to 507, but much better selectivity. So we're on the order of 100-fold selectivity with that compound. So as of today, we have not tested this new compound yet. I'm looking forward to getting the opportunity to test this in mice, particularly with our progressive ratio assay, where we can do repeated tests in the same mouse and gather information that I think will be useful for thinking about clinical uses. Unfortunately, we had to rederive the mouse line, and we've had some difficulties with that process that have delayed things a bit. But we hope to be able to test LI967 in the near future and continue developing this program. And so with that, I'll thank the many people who've worked on this project within the drug discovery group at the Lieber here on the left. And also thank our collaborators at Lieber and within NIH who've worked on the KCNH2 project with us. And so I believe we have plenty of time here, so I'd be more than happy to take any questions you have.
Right. Thanks, Greg. Thanks for like a really interesting presentation. It's great. It's great to see like you know, a really good attempt to translate human genetics into a sort of a mouse model directly. So yeah, so I've got a few questions. So you know, in terms of the impact of your 3.1 variant on CNS function, have you looked at broader battery? You know, is it only things like motivational deficit? Have you tried things like rotor-rod? Is it a specific effect, or is it a more widespread CNS depression?
Yeah, so from a behavioral perspective, it seems to be a very specific set of behavioral tasks. So as I mentioned, we did a large battery of tests with these mice. So right. Just from a gross perspective, they look normal. So movement, no sort of motor impairments that we could see. Locomotor activity in an open field is completely normal. Fear conditioning, normal. As I said, object recognition, and even on the T-Maze task in sort of the easy settings we use for training, they're relatively normal. We see deficits in very specific circumstances. So as I said, when we tax their working memory capacity, and what's really important with that task is it's the spatial component. So we think it's the crosstalk between the prefrontal cortex and hippocampus that's critical there. And there's been some subsequent work from Feng Yang who's shown that these mice actually have pretty severe deficits in hippocampal prefrontal synchrony. And so we think any sort of task that requires that real-time interplay between prefrontal cortex and hippocampus, these mice are going to struggle with. And tasks that engage some other systems, they may be able to compensate.
Right. Okay. And okay. So is that why? Because obviously the 3.1 variant would be expressed across quite large areas of the brain. So that's your explanation why it's so specific, because there's compensation in other parts of the brain.
We believe so, and we think just sort of there's a much smaller margin for error in terms of long distance signaling in these regions, that with the lack of synchrony, you're seeing that play out in behavior. In some other areas, there may be a bit more leeway if the timing of a signal isn't exactly where it needs to be.
Okay, cool. Right, and yeah, what about so turning to your pharmacology? I mean, that looks really exciting that you're getting such selective compounds, and I guess you've screened them against other potassium channel subtypes. Are they truly selective?
They seem to be truly selective. Yeah. So we haven't done sort of a full panel of screening, but for sort of the near neighbors that you would think of, they are fairly selective.
Right. Okay. And you know, the obvious question is, you know, because obviously the poor domains conserved across 1A 3.1. You know, what's driving the selectivity? You know, it seems surprising you've managed to achieve such selectivity.
Yeah. No, that's a great question, and I think you know, getting some structural information here, I think would be very helpful as we move forward with you know, some of the SAR around these lead compounds, to really get a sense for where are they interacting on the channel, because as I showed, there isn't a huge difference in structure in terms of the number of amino acids that are different, and as you said, the pore region is fairly conserved. So it will be important for us to get that structural information on where our selective compounds are binding with respect to, you know, some of the non-selective blockers that are out there.
Okay, brilliant. And what about you know, I guess the key thing is what is the target? Because obviously, you know, that there's so there's more than hERG1 expressed in the cortex. There's hERG2 and hERG3. So is it forming heteromultimers or is it a homomer? What are your thoughts on that?
No, that's a great question, and so we've focused on profiling on two different levels that are really at opposite ends of the spectrum. So we've profiled in, you know, these cell-based systems where there's only expression of 3.1 or 1A, and so, you know, the assumption would be those have to be homomers. And then on the other end, we've looked at intact animal systems and behavioral outputs, where we're measuring sort of the conductance that's the result of, you know, any number of channels and any number of heteromers, potentially homomers. So we don't have the information yet on whether or not in an intact system 3.1 is, say, forming only homomers. So you have 3.1 homomers, 1A homomers, and you can think of the other potassium channels that may be coming into play here as well. Or if there's heteromers forming, right? So two 1As and two 3.1s or whatever the mix may be. So I think that'll be an important feature to uncover, so we have some insight into what's actually happening. As I said, we've been really focused on just the output, which is the final product of the interaction of a number of different ion channels and ion channel subunits. So that's an outstanding question, an open question that I think we'll need to answer.
Yeah, and what about translation to primate? Because is your overexpression similar to the levels in primate, or is it a super overexpression? So I mean, how do you get the translation right?
Yeah, that's another important thing for us to think about. So it's most likely fairly close to what you see in terms of ratio to the people who carry the risk SNP that's associated with increased expression of 3.1. One of the key features, I think, is with respect to the heart, even 1A is very low expression in the brain. And so what we're really seeing is, on top of sort of a baseline of low 1A expression, we're getting much greater expression of 3.1. So I think our transgenic model, has the ratio correct in terms of 1A to 3.1. My guess is we're probably getting a little bit more total KCNH2 expression. So it is a bit higher, but I think the ratio is probably what's more important and what's driving the function. And there, I think we have a pretty good measure of the risk associated allele and the situation that's creating in the brain.
Okay, brilliant. Okay. Yeah, so one of the questions is, how did you find these? How did you find the selective blockers? What did you screen? What was your screening subset?
Yeah, no, that's a good question. I don't know offhand the screening subset. So that was part of a collaborative project with NIH and the NCATS group at NIH there. So it was part of a screening collaboration with them.
Okay, brilliant. And are these the only compounds? Have you found compounds with sort of different state dependencies, different characteristics, or what sort of compounds have you identified?
Yeah, so these are the main compounds that we've identified with the selectivity. So I don't have much information on sort of state dependency, if they're particularly which stage of sort of channel activation they may be preferentially binding to. So I don't have much information there.
Sure. I guess it's are they relatively rare? Because to find selective blockers within the same with just a splice variant, I guess the number of inhibitors you found that were selective was pretty small, was it?
Yeah, it's not a huge set of compounds that show sort of this appreciable selectivity. That's true.
Okay, brilliant. And the other thing is another suggestion. To develop a successful small molecule might be challenging because the amount of substrate you identify will be small. Is a Crispr approach a better therapeutic option? Yeah,
so I think it's a possible therapeutic option. I think that may be difficult, even given the valid concern you've brought up about a small molecule. I think possibly the difficulty with a CRISPR approach is more than likely 3.1 is playing a functional role under normal conditions, and so we probably don't want to get rid of this isoform completely, because we think it probably has stuck around in primates because it serves some important role. You can think of sort of diversity of the firing properties of neurons, and adding to that diversity may be a good thing for an organism. There's some work that's suggested, some computational work that suggested that KCNH2 is important for regulating dopaminergic neurons and the different firing patterns that they're able to produce. So I think a CRISPR strategy may be difficult because you would be getting rid of even sort of the advantageous aspects of hERG processing. So we're hoping we can kind of just tamp down things a bit, but there's even the difficulty with pharmacology, and you don't have sort of the resolution to just tamp down, and so that's part of the reason why we're focusing on the electrophysiological activity as a readout, because if we have sort of a pathological EPhys signature that we can bring back into the normal range and we can titrate dose and do that, that'll give us kind of I think a good readout to see if we're having therapeutic effects but not having detrimental effects of inhibiting 3.1 too much.
Right, okay, yeah. So do you think, I mean, it is, looking at the sort of, looking at a cellular level in terms of excitability, do you think, what is the pathology? Is it just increased excitability generally, or is it a disruption of the spike frequency adaptation? What do you think is the?
My thought is that it really sort of changes the timing of ensemble activity within a circuit.
Right.
And so, I'll go back to, what we know about the T maze and performance in the T maze. You really need precise synchronization between the hippocampus and prefrontal cortex. And a lot of this work has shown in sort of theta frequency bands. You need precise synchronization. And so if you have a small subset of neurons, who are returning to base, they fire an action potential, they return to their resting potential, a split second too fast, you've now thrown this circuit out of whack from a synchrony perspective. So I really think that's sort of the pathological substrate that we're dealing with here, is that the timing of the circuit is just off a small amount, and these subtle changes are leading to sort of breakdowns in behavior.
Right. Okay. And are you going to investigate that further? That hypothesis?
We would definitely like to, and so that's part of the reason why these tasks where you can sort of have discrete trials that you can repeat. And so it's really one of the reasons why I'm excited about the progressive ratio work, because there you can get hundreds of trials. The behavior is very stable. So mice tend to day after day produce sort of the same level of output. And so there I think we can really answer those questions around, what's the precise effect that is correlated with a change in behavior, and can we bring it back into the normal range with a pharmacological intervention, and really work out that relationship, the PK/PD relationship there.
Right, fantastic.
Hi everyone, thank you for attending today. Oops, I'm not on the camera. There we go. So yeah, thank you everyone for attending, and thanks Eddie for that Q&A, and thank you first and foremost to Greg for a wonderful presentation. Very informative, very interesting as always. Great to hear from you today, and thanks again for offering to present for us today. It was really fantastic. Our next speaker is to be confirmed. We have a speaker, we don't yet have a date, so we're going to announce that very soon on our website. So please look out for it coming up, and we'll also be advertising it through LinkedIn and through our usual channels. Yeah, thanks everyone for joining, and we're going to end there. Thanks again to Greg, thanks to Eddie, and we hope to see you all on the next webinar. Thanks.
Fantastic. Okay, thanks Greg.
Take care. Bye-bye.
Bye everyone.